All right. I know there was some confusion last time about the location. We're going to stay here, so if you see other people that are still confused about the location, please let us know. Oh, that's the volume? Great, thank you. We're trying to change that in the Canvas website as well. In case you weren't here last time or you didn't get to sign a consent form about the filming of the lectures, please do so. Talk to Nimrod. We're going to be trying to post slides, as well as we're asking every person who will be giving a lecture to propose one paper to read, one recent work in the field. So that's going to be posted in the website as well. I think that's partly in the modules section, and also Nimrod, is that in another section as well? The reading material?
Yes, there are two sections. One is the reading material on the website, and also in the modules, where you can see all the lectures. There are also suggested papers by all the speakers or something that you better read before the lecture.
Yeah. And then we're posting the slides as well, and then 1 or 2 days after lecture, hopefully we'll have the videos as well. So today I'm going to talk about vision, specifically visual recognition. And the study of vision has played quite a fundamental role in our understanding of intelligence in general. We hope that if we can make progress in building intelligent machines in the context of vision, we are going to be simultaneously making progress in many other domains, and that some of these ideas will extrapolate to many other aspects of cognition. We're interested in vision because it plays such a predominant role in our daily lives, because of multiple applications in the real world, because of our access to stimuli, to images, to controlling stimuli during experiments, and also because we can stand on the shoulders of previous giants who have examined the inner workings of the brain during visual recognition. And I'll give you a brief summary of that work today. What's particularly amazing about vision is that we can capture a very large amount of information in a very short amount of time. If I were to try to tell you everything that I'm seeing here, it would take me a very, very long time. An image is worth more than 1,000,000 words. So this ability to convey a lot of visual information very fast has probably played quite a predominant role in the evolution of the visual system. So we can take an image and we can answer essentially an infinite number of questions about that image. Who's there? What is there? What are they doing? We can even make inferences about what happened before, what's going to happen next. We can search for specific objects. We can also make inferences about actions and many other questions. So ideally, passing the Turing test for vision would mean that we would be able to take any picture, any arbitrary image, and answer any question that a human would be able to answer on that image. And if we can really solve those types of problems, I think we would have made a lot of progress also in terms of being able to solve problems of in the clinical world for diagnosis. I referred very briefly last time to self-driving cars, security applications, building artificial vision systems for the blind. And for me also, one of the main goals is to be able to understand the brain, to be able to understand how the brain processes visual information. So this ability to convey a lot of information very fast probably played a central role in the evolution of the visual system. But also, Andrew Parker, a zoologist at Cambridge University, put forward the idea known as the light switch theory, the idea that the evolution of animal species largely was triggered by the apparition of vision in the world. This is partly because the first eyes came about 500 million years ago, which coincides with the so-called Cambrian explosion, the very rapid expansion of the different types of animals in the world. So the idea is that by being able to capture visual information, the ability to begin to interpret patterns, that gave rise to an arms race between prey and predators, and to the rapid explosion in the animal kingdom. Fast forward many years, that gave rise to the possibility of navigating, of building social interactions based on vision. Even the preliminary rudimentary forms of recognizing patterns in the stars, and therefore initial steps towards astronomy. And that also played an important role towards being able to predict seasons. And with season prediction came agriculture and changing from a nomadic lifestyle to a more sedentary lifestyle as well. So to some extent, a lot of these major transformations throughout evolution can be traced back to the very rapid ability to convey visual information. And that's the essence of this light switch theory by Andrew Parker. Any system capable of doing intelligent visual understanding must satisfy three fundamental properties that I'd like to highlight. The first one is selectivity, our ability to discriminate images that are very similar and being able to tell them apart. One example of that is face identification. Faces are relatively similar in terms of pixels, and yet we can tell them apart very well. Another example is our ability to recognize symbols, which was probably fundamental to the beginning of language. Just memorizing, just achieving selectivity is not enough. So one way of achieving very high selectivity is to memorize pixel by pixel what's happening in the image. And that doesn't work, and that doesn't work because it doesn't have the second property, which is invariance. That is the ability to recognize an object despite the infinite transformations of that object. Any object can cast an infinite number of projections onto the retina. Those are transformations in terms of color, in terms of scale, in terms of rotation, in terms of occlusion, and so on. And we want to be able to recognize those objects despite all of these transformations. So one of the central challenges in visual recognition is the combination of exquisite selectivity with invariance to transformations. And all of this needs to happen very fast. So without telling you exactly what to expect here, I'm flashing a lot of random pictures here, depending on the speed of my computer and that projector. Each of those images probably lingered in your retina for about 100 milliseconds or so. So there were about 10 pictures per second. And yet you probably would recognize many of those images very well. So in about 100 milliseconds of processing, your brain was able to recognize complex images without any prior expectation, without even knowing what I was going to show you. You don't have to solve fancy differential equations to do vision. You don't need to change and turn on different genes that take minutes to do vision. In 100 milliseconds, in a very small fraction of a second, you can capture a lot of visual information. So we need to be able to explain these three fundamental properties. So starting from the very beginning, the reason we can see is because objects reflect light. The reflected light goes into the eyes, into the retina. To a first-order approximation, we can think of the retina as a very fancy and very sophisticated digital camera that outputs pixel-like information to the rest of the brain. The retina does much more than that, but that's a good starting point to think about visual processing. If we think about the sort of type of light that impinges on the retina, we could think that there is a very large number of possible patterns that we would have to recognize. For example, consider a patch of 100 by 100 pixels. And just to simplify, let's get rid of color. And let's imagine that each pixel has 256 shades of gray. Okay, so in a colorless world, we're looking at a very tiny fraction of the world, just 100 by 100 pixels. Just the number of such images is enormous. Okay, so if there's only one pixel, there are 256 possible images. If there are 100 by 100 pixels, this is the total number of possibilities. So how could we ever learn to recognize objects given this enormous astronomical number of possible images? And part of the answer to that is that the natural world, the kind of images that actually reach the retina, are very special. Only a very tiny fraction of all possible images are the kind of images that we encounter in the world. One of the most striking aspects of the natural world is that the natural world is roughly continuous and smooth in space. So for example, if you take an image like this one, and you look at the pixels in a given position, and you correlate with those with the intensity of the pixel in a nearby position, you see that there's a very strong correlation. So nearby positions in space are strongly correlated. So roughly speaking, the world is very smooth, with a few discontinuities given by very rare but very important edges. Edges are discontinuities and places where there's a major transition in intensity. But by and large, most pixels are very correlated with the ones next to them. In addition to this spatial smoothness, there's also a temporal smoothness in the world. By and large, the world is quite static. Objects move in the world. We move our head. But those are pretty rare occurrences. Most of the time, things don't change too rapidly in the world. In fact, one of the main changes in time is given by eye movement. So when we're looking at an image like this, we're constantly moving our eyes. So this is an actual video tracking the eye movements of a human subject during the course of 12 seconds while they're examining this image. If you've never reflected about eye movements, you may not know that you're moving your eyes all the time. We're moving our eyes about 3 times per second, more or less. These are the large saccadic-like eye movements. In addition to that, there are very, very tiny, smaller eye movements that we make even faster at an even faster rate. And you typically don't know. The world seems pretty stable, despite the fact that we're constantly moving our eyes, even about 3 times a second. But that means that for about 200, 300 milliseconds, the world is approximately constant. So there's a temporal smoothness and there's a spatial smoothness as a major constraint to the world. Vision starts in the retina, and there has been beautiful, elegant work to try to establish the anatomical and physiological pattern of cells and connections and computations that happen at the level of the retina. So this is a diagram of the major cell types in the retina. In this diagram, light comes actually from the bottom, from the bottom of this image, and lands on these so-called photoreceptors. Photoreceptors do something that's quite extraordinary, which is that they convert energy in the form of light into electrical signals that will be then sent to the rest of the brain. So this ability to convert light signals into electrical signals is probably one of the fundamental steps in evolution to be able to give rise to all the amazing things that we can do with our eyes. These photoreceptor cells come in 2 main varieties. One is called the rods, the other one is called cones. The rods are the main types of receptors that enable us to see at night when there's low intensity. And the cones are the main type of receptors that enable us to see color. And they're also concentrated in a part of the retina called the fovea, which enables us to see in high resolution. The activity from these photoreceptors is conveyed through this cascade of cells into these cells at the bottom here, which are called retinal ganglion cells. The retinal ganglion cells are the ones that produce the output of the retina that conveys information to the rest of the brain. The responses of retinal ganglion cells can be approximately described by this difference of Gaussian operator here. So what you're seeing here is the magnitude of the response of a typical retinal ganglion cell, specifically what's called an on-center retinal ganglion cell, one that is activated by input in the center of the receptive field as a function of position. The receptive field is the particular location in the visual field that the neuron responds to. The fact that we can see essentially throughout a field of about 180 degrees means that there have to be retinal ganglion cells tiling the entire visual world with a particular receptive field in that location. So these receptive fields of retinal ganglion cells have 2 components. One is an excitatory component given by this red Gaussian here, and then a shallower and wider inhibitory component. Because of this interplay between excitation and inhibition, if you're looking at something completely smooth, like this wall over here, where there is no change, and there is stimulation of the center of the receptive field, but also the surround, there's surround inhibition. So the inhibition from the surround essentially compensates for the excitation, and the responses of retinal ganglion cells are significantly reduced. The type of stimulus that excites a retinal ganglion cell is when you have light in the center and essentially no light in the surround. So these cells are essentially capturing some sort of spatial gradient or difference in the image between what's happening inside that receptive field and what's outside. So the cells are interested in spatial change. The cells are also interested in temporal change. When when you flash a stimulus or when there is something moving, that's also a very important component of activation for retinal ganglion cells. Any questions so far? Okay, so the information then is reflected on objects. It goes to the eyes, and then from the eyes, it goes back. It goes to the rest of the brain. And ultimately, what we're seeing is correlated with the magic transformation of this pixel-like representation into our perceptions. And that happens through a very rich and complex cascade of processes that we're only beginning to understand, and that is schematically illustrated in this diagram here that was put forward by Feleman and Van Essen a few decades ago. So here at the bottom of this diagram is the retina. These are the retinal ganglion cells. And each one of these boxes corresponds to a particular part of the brain, a particular area of the brain that's responsible for processing visual information. In our brains, as well as in other primates, a large fraction of the brain is involved in processing visual information. There are tens of different areas that are involved in processing visual information. In particular, here are the retinal ganglion cells. The information goes to a particular location called the thalamus, the LGN. And then from there into the back of our brains, into primary visual cortex, and then from then onto a cascade of different cortical processes that transform visual information from this pixel-like representation into our ability to answer a large number of questions. What we end up perceiving, our perception, is a construct. Our brains are essentially making up stuff all the time. And this can be illustrated quite prominently in a large number of visual illusions, many of which you're probably familiar with. This is a classical visual illusion. So if you look at this, this line here appears to be longer than this one. That's a pretty strong visual illusion. I'm not cheating. So if you put lines here, you can see that they are actually exactly the same length. Yet your brain is making you think that one is longer than the other. Another example of perception as a construction, as an invention of something that your brain is making up, is given by the blind spot. It turns out that if you close your eye, there's a particular part of the visual field that doesn't map onto any photoreceptors. And yet we don't see a black hole in the visual field because our brain makes up the rest. Our brain essentially has a filling-in process so that it interpolates. And if you've never seen the blind spot, there are multiple demos online. You can just Google this, and there are multiple demos online that help you try to localize this blind spot. I want to quickly refer to some basic aspects of perception studied at the psychophysics level and how we put together different pieces of information to group objects or to group lines into coherent wholes. So this is one famous visual illusion or example. It's known as the Kanitsa triangle. We see a very strong illusion of a white triangle here. That means that we think that there are white lines, white edges here that are part of this triangle. There's absolutely nothing in there. This is just our brain, again, making up stuff. And this is what the Gestalt psychologists refer to as the law of closure. We like to, our brain tends to complete shapes and put together lines to make a whole. Another similar essential law of grouping and perception is the law of similarity. When we look at a picture like this, people tend to describe it as a line of these black squares, another line of these white squares. And this has been capitalized in many pieces of art where we try to essentially we tend to group things that are similar. Another rule that we use to group objects is by proximity. When things are close, we tend to think that they go together. For example, this is usually described by most people as a collection of vertical lines rather than horizontal lines, and the converse is true here, again, due to proximity. And this, again, is something that people capitalize on all the time to make these kind of funny pictures like this one based on this notion of proximity. Another thing that people tend to do is to continue lines. Therefore, your brain may think that this hand here corresponds to this person because of this continuity here. Of course, that's not the case. In order to begin to investigate what are the brain processes that are responsible for these illusions, that are responsible for this ability to convert pixels into perception, we need to figure out which parts of the brain to study. And one of the main clues that usually has served as a starting point to figure out brain mechanisms of perception is the study of lesions. People who have lesions in their brains, either due to genetics or due to accidents or due to virus infections or a myriad of other conditions. People may have damage, they may have lesions in different parts of the brain, and that may lead to specific deficits in visual perception. Just to give you one example of those studies, there's a famous book by a British writer called Oliver Sacks, The Man Who Mistook His Wife for a Hat. This is actually quite literally what happens in a very rare condition known as prosopagnosia. Prosopagnosia refers to people who have an inability to be able to recognize faces. They can still see light, they can see color, they can discriminate different shapes, but they have a very specific inability to recognize faces. And this particular patient that Oliver Sacks studied had damage in the anterior parts of the temporal lobe and was inability to distinguish between a hat and his wife. So this is a pretty striking visual deficit. And there are many such deficits that have pinpointed the way towards what are the functions of all those boxes that I alluded to in this type of diagram. So this type of diagram is based on anatomical connections, injecting a virus in a particular cell and then, or a tracer in that particular cell, and then tracing the connections, so seeing which particular location in the brain is a retinal ganglion cell projects to, and similarly for primary visual cortex and so on. So this is the way in which they determine a lot of these different brain regions. And then lesions provided fundamental insights to begin to study these regions in terms of what kind of visual deficits are caused when one of these boxes is essentially deteriorated. The next step after studying lesions, now you know that a general area may be involved in recognition of faces or in processing colors or other attributes of the image. So to really elucidate the types of circuits and computational mechanisms involved in visual processing, what biologists tend to do is go into the brain with electrodes to record the activity of individual neurons and correlate the activity of those neurons with different aspects of the visual stimuli. So I'll give you just a brief flavor of the type of responses that people have described along visual cortex. The first observation I already mentioned last time. All of this line of research in trying to elucidate how visual cortex processes visual information can be traced back to the elegant work of Hubel and Wiesel, who got a Nobel Prize for recording the activity of individual neurons in primary visual cortex and realizing that neurons are tuned to lines of particular orientations. So in a similar vein, people put electrodes essentially in many of these boxes, so you can go into each one of these areas, put electrodes, and listen to the activity of individual neurons in those areas. That is, record the neurophysiological responses upon presenting a variety of different stimuli. So one of the first things that you observe if you do that is that the latency of the response essentially increases progressively from one box to the next. So this is the particular part of the thalamus that I mentioned, the LGN, which is the first stage in processing after the retinal ganglion cells. There are two flavors of the LGN. These are described here. This is a cumulative distribution. This is the percentage of neurons that have a certain latency after stimulus onset. And so basically, what you see is that there is a progression in the latency of the responses as you go up in this diagram. So when you go from here to here to here to here and so on, it takes a little bit longer for the neurons to respond. By the way, most of the time we're not aware of these latencies. That is, it takes time for information to reach a particular part of cortex. We tend to think that vision is instantaneous. You close your eyes, you open your eyes, and the world is out there. But it takes a little bit of time for information to propagate from the retina all the way to the highest echelons of visual processing, which are ultimately responsible for your perception. So essentially, everything we see has happened in the past. Not far in the past, about 100 milliseconds or so in the past, but it has happened in the past. So what we're seeing is a rendering of what was out there in the world about 100 milliseconds ago. Because of this progression here, combined with this type of anatomical diagram, people have made the observation that as a first-order approximation, we can think about processing in cortex as a hierarchical process. Information goes from here, then to here, to here, to here, to here, and so on. You can see from this diagram that that's only an approximation. For example, there are some neurons, let's take this area. This area is called area V2, and this area is called area V1. So there are some neurons in this stage, which is V2, that fire before neurons in V1. So it's not a perfect relay race where you actually have to wait from one stage to go to the next. But on average, it takes about 15 milliseconds or so to go from one visual area to the next. So there are about 15 milliseconds of computation in each one of these stages, such that at the end of the day, in 100 milliseconds or so, we are able to do visual recognition, as I briefly illustrated when I flashed a lot of pictures to you. Another observation that was made by recording the activity of neurons in different brain regions is that if you plot the location of the receptive field of a given neuron and the size of the receptive field as a function of the location, for any given area, for example, V1, there is a progressive increase in the size of the receptive field as a function of the position of the receptive field. So remember, the position of the receptive field is which part of the visual field the neuron is responding to. So this area here, near the center of fixation, that is where you have your eyes, that's the fovea. And as you go farther and farther away within any given area, the receptive fields become larger and larger. So that means that you have less and less resolution as you go away from fixation. And because of this, whatever we see in the fovea it has very high resolution, and whatever we see in the periphery is blurred and has much lower resolution. We have the illusion that the entire visual field has the same resolution, that everything is perfectly clear throughout the visual field. And if you've never stopped to think about it, you may not believe me that everything outside of your visual field is blurry. Perhaps one of the best ways to see that is when you're trying to read, when you're reading text. Make sure you don't move your eyes. You fixate on a given letter or a given word and try to read a word that's in the other page. Okay? It's basically impossible to read outside of one or two words near exactly where you are fixating. And this is because of this drastic change in resolution. The reading requires high resolution, and we can only do that wherever our eyes are. In addition to this increase in the size of the receptive field within an area, there's also a progressive increase in the size of the receptive fields as we move from one area to the next stage of processing in the visual field. So this is a diagram illustrating that idea that within an area, receptive fields are larger towards the periphery. So this is the fovea and then the periphery. And then across areas, also the receptive fields become larger and larger. Yes?
How do they get the size of the receptive field? Is it behaviorally?
This is physiologically. So all of this is based on physiology. So you're recording the activity of a neuron. Okay? So you're measuring firing rates, how many times the neuron fires per second. And then you show light in a given location. And let's say that the animal is fixating. So I'm fixating in a given location. You shine light here, nothing happens. You shine light here, nothing happens. Until you suddenly discover that when you put light in this location, the neuron goes crazy. The neuron is activated so that's the location of the receptive field. And then you can play with the size, and you make it larger, you make it smaller. If you make it very large, you have these surround mechanisms that I briefly described in the retina. This idea that the surround is inhibitor. So if you make the stimulus very large, the response of the neuron decreases. So the size of the receptive field is essentially the area that maximally activates this neuron, and that's how they come up with this axis as a function. With these numbers as a function of the location. Yes?
One other thing was, on the previous slide, you were saying how like V2 responds sometimes, like some percentile responds earlier than V1. Does that sort of be like feedback connections maybe, or?
Yeah, so that's a really good question. So again, this is just a cumulative distribution over many neurons, and we don't know exactly the entire pathway that activated that particular neuron. So I think what you are conjecturing, and it may be the case, so let's take V1, for example, here. Some neurons in V1 respond very, very rapidly. Others seem to be lazy, and it takes longer for those other neurons to respond. Why is that the case? So part of it is maybe the different thresholds on different neurons, the different sensibility to stimulation. But you're arguing, and it may be that some of these neurons may be in different layers. In V1, there are multiple layers, and some of them may receive more input from bottom-up sources like the LGN. Some of them may receive feedback signals from V2. So I think we're beginning to examine many of these questions. And here, all of that is put together. This is a cumulative distribution over many neurons in these areas. But it's a very good question. Any other questions, comments? Yes?
Does this also apply to reflexes, or is this just regular vision? It seems like my eye remains closed, and I think it might be less than 0.1 seconds. I think so.
So the way this is done is essentially you're recording the activity of a neuron, and then at time 0, somewhere here, at time 0, you flash light on the center of the receptive field for that neuron, and then you measure how long it takes until the neuron becomes activated. Okay, so these are essentially latencies of responses to flashes of stimuli. Did that answer your question? Or so, and in many of these experiments, just to talk about blinks, you're ensuring that the animal is fixating, that the eyes are open, and that they're not moving. So if the animal is moving in the eyes, then all of this may change if you're blinking. So all of those trials are discarded. Okay, but
I think I'm referring to the situation where like an object responds towards my face. So like I would have reflexes with my mouth closed. I was under the impression that that happens a bit faster than 0.1 seconds.
right. So all of this is referred to a situation where there is no stimulus, and then a stimulus appears and is being flashed. So, but a different question is, an object is looming towards you, okay, and how fast can you react to that information? So, embedded in here, particularly when things are moving, there's a pathway that can detect motion quite fast. So there's a division of labor in the visual system. Basically, there's a so-called what pathway and a where pathway. And one of these pathways is particularly relevant for visual shape recognition. The other one is particularly relevant for detecting motion and object locations. As your intuition correctly inferred, the pathway that's involved in detecting motion, things that are looming towards you or things that are moving in the environment, that actually works faster. And that's actually the distinction. I didn't get into those details, but that's the distinction between these and these here. So these are both parts of the LGN. It's part of the same brain structure. And then you can see that there's a pretty large difference. These cells are more interested in motion, okay? And they respond much faster than these cells here. And similarly, that happens in other parts of cortex as well, okay? So that's a good point. So motion can be detected faster than a lot of the questions about shape. Okay. So I won't have time to do justice to the last 3-4 decades of recording activity throughout visual system. In the retina, Stephen Kufler was one of the pioneers in describing the receptive field structure of retinal ganglion cells. I mentioned the Nobel Prize winning discovery of Huvel and Wiesel. If you put electrodes in this is what I was referring to as the what pathway and the where pathway. This is particularly relevant for shape recognition. This part of the brain is more relevant for recognizing object locations as well as motion. So if you put electrodes in part of the parietal pathway, there are neurons that are very sensitive to motion. They may respond to a bunch of dots that are moving in one direction but are not in the other direction. So there are mechanisms that are instantiated to recognize motion. Whereas if you put electrodes in areas along the ventral visual stream, particularly in the highest echelons of visual cortex called the inferior temporal cortex, there are neurons that respond selectively to complex shapes. So here's a neuron that was described by Bob Desimone and Charlie Gross. What you're seeing here, these histograms correspond to the degree of activation of the neuron. So this is a neuron that was particularly activated by pictures of monkey faces more so than hands and other shapes. So there are neurons that have pretty fancy tuning curves and respond to complex shapes. So just to give you, I mentioned earlier that everything we see is a construction. Our brains are making up stuff inspired by and dictated by what's outside in the world. But ultimately, the brain is the one that's constructing our interpretation of the world. If the brain is doing that, there have to be neurons that do that. And indeed, here's an example of neurons in this area called V2 that responds selectively to these illusory contours. So for example, your brain is making up a line here when there is no line there. Well, it turns out that in this area V2, you can record the activity of neurons that essentially are firing and telling you, yes, there is a line there. So basically, you can put this part of the visual field exactly in the center of the receptive field for the neuron, and you can record the activity of the neuron, and the neuron gets very active saying, yes, I see an edge here. So there are neurons that are seeing those imaginary edges and making this, and fundamental to make this construction of the world. Most of what we know about recordings, everything I said so far, comes from recordings in animal models. In a few rare instances, it's possible to also examine the activity inside the brain in human subjects. This happens in patients that have epilepsy. It's illegal to go and poke electrodes inside a human brain unless there's a particular clinical condition to do so. So in patients that have pharmacologically intractable epilepsy, neurosurgeons may want to try to resect the part of the brain that's responsible for the seizure. So these are patients for whom none of the known drugs work in terms of stopping seizures. And so the neurosurgeons try to put electrodes to localize what's the origin of those seizures. And while the patients stay in the hospital, it's possible to record the activity of either neurons or ensembles of neurons and ask properties about visual recognition inside the human brain. So this is an example of the type of electrodes that are placed. These are locations of those electrodes inside the human brain. And here's an example of recording from one of these electrodes in this area called the inferior temporal cortex. This is one of these areas I just showed you, one neuron from monkeys that was responding selectively to complex shapes such as faces. And what you're seeing here is an intracranial field potential signal. This is a measure of the average activity of many, many neurons in the vicinity of the electrode. This is the activity, this is the field potential signal as a function of time when different pictures were presented for 200 milliseconds. So each of these colors correspond to different types of pictures. This one in particular here corresponds to faces. So this is an electrode in the human brain that responded selectively upon presentation of pictures of faces, similar to the kind of studies that I alluded to in monkeys. So there is a rather strong parallel between what we understand about visual cortex in animal models and those few cases where we can study these properties in the human brain. If you go beyond visual cortex, In this case, these are the targets of the visual system. Here's another example of a recording of single neuron activity from the human brain in one of these patients. And what you're seeing here are the responses of one neuron. All of this is one neuron in response to each one of these pictures. Each one of these marks here, each one of those ticks corresponds to a spike or action potential from that neuron. There are multiple repetitions of that picture, and these histograms correspond to the average activity of that neuron in response to that picture. So you can see that this is a neuron that, like many other neurons, it fires spontaneously many times, so there are a few spikes here and there. But the neuron was particularly interested in the presence of pictures with this actress here called Jennifer Aniston. So this became known to many people as the Jennifer Aniston neuron. This is a neuron that shows a pretty striking degree of selectivity. It's not just responding to any phase, so it's different from the kind of neurons that I showed in the previous slide. It's not responding to any phase. It's very selective, and yet at the same time it's quite invariant. That is, it's responding to pictures that are very different from each other. This is probably not vision anymore. This is an area that's outside of the visual system, and we can debate about the origins of what's exactly represented by this neuron. Yes? This is a single neuron. This is one neuron. This is not. This is an ensemble of many, many neurons. Probably 1,000 neurons or 10,000 neurons are probably part of this ensemble activity. This is one neuron. Okay, so these are patients that have epilepsy. The neurosurgeons implant electrodes in order to localize the seizures. And in this particular case, they put a 40 micron high impedance electrode. It's a very, very thin, very small electrode. These are extracellular recordings. That means that the electrodes end up being outside of the neurons. But based on the proximity of the neuron of the electrode to the neuron, and based on the characteristics of the extracellular field potential, it's possible to ascertain that in all likelihood we're talking about a single neuron as opposed to cases where we have multiple neurons. So this is similar to the problem. Sometimes this is referred to in a different domain as the cocktail party problem. Imagine I have a microphone here, and there are lots of people talking at the same time. So this is an electrode and multiple neurons, right? So there's a microphone, many people are talking, okay? So in principle, you could be recording the voice of every single person here. But if you put the microphone really close to one of them, so the microphone ends up being very close to me in this case, even if a lot of other people are talking, the microphone will mostly capture my own voice, and my own voice will have a much louder volume. So same thing happens when you're doing extracellular recordings. So if the electrode is sufficiently close to a neuron, all the chatter from other neurons that are farther away is hard to capture. And that's how people ascertain that this is a single neuron. Yes?
How does this neuron react in people who don't know who Jennifer Aniston is?
OK. So inside story. In this case, many of the investigators didn't know who Jennifer Aniston was. But this particular patient actually did know who Jennifer Aniston was. Not only that, she knew Jennifer Aniston personally. This was a recording in LA. And so we think that this type of highly selective, highly invariant responses is probably only happens in cases where people are very familiar with those stimuli. It's unlikely that people have such a sparse representation of information for all possible objects or all possible phases or all possible stimuli. So I think it's a good question. I think you're onto something. It's quite likely that that this type of representation only happens for things that are very familiar to the person who has this neuron. Yes?
Coincidence that, like, when the sperm comes out in position of a bunch of pictures and it happens to respond mostly to Jennifer Aniston, or was that coincidence that they happened?
So this is in many of these experiments. Basically, the investigators don't know a priori what the neurons will or will not respond to. So this is a fundamental question in visual experiments. You need to decide what kind of pictures to present. A lot of the big findings in vision have actually happened in a rather serendipitous fashion. Hubel and Wiesel discovered orientation tuning, and the reason they discovered that was because they were studying center surround receptive fields. And in those days, you're probably too young to know this, but they had slides. They had these things that you put in a projector. That's how they presented visual stimuli. And after doing an experiment with circles, they realized that whenever they put the slide into the projector, the neurons started firing like crazy. So similarly, the neuron that I showed you from Bob Desimone, they were presenting all sorts of stimuli, and all of a sudden they realized that the neuron from this monkey went like crazy whenever one of the investigators passed in front of the monkey. So that's how they discovered that some of these neurons responded to faces. So in a similar fashion, in many of these experiments, people essentially tried a large battery of pictures and hoped that some of the neurons would be interested in those pictures. Subsequent versions, partly inspired by the previous question, try to ask patients what they like or what they are familiar with, what kind of TV programs they watch, in the hope that some of those answers might help find neurons that have this type of selectivity. But by and large, there's a lot of serendipity and randomness in the decision of which pictures to show. This is just one example. Most neurons actually do not respond like this. If you actually show most neurons, this would be completely flat lines and not very exciting. In most cases, it's very hard to find what the neurons are actually interested in. These are just a handful of examples that respond in this way. OK. So I want to. I've been mostly talking about brains and neurons. Now I want to switch to computer models and computational models of the visual system. So just to emphasize again what the problem is. Does anyone know what this is? OK. So it's very hard to. I would be very surprised if you, unless you've seen me talk about this before, because I've used the same example. It's very hard to decipher what this. But this is basically what vision is based on, right? So we can imagine, at least to a very good first approximation, retinal ganglion cells that are sending information to the rest of the brain. Each of those cells is sending a number of spikes per second, a certain scalar number, which is a firing rate. Okay, and the brain has to decipher what's out there based on this type of representation. So that's what this picture is. So what you're seeing here is actually this part of that this part of the image. Okay, so this is part of a flower. Okay, so it's essentially impossible to see that in here. It's of course very easy to see that in the actual picture. Yet your brain is essentially capturing a set of numbers. That's what vision is all about, right? It's about retinal ganglion cells that are firing. There was a transduction of light signals into firing rates, and your brain needs to be able to take this kind of matrix and make you think that you just saw a flower. Okay, so that's what we want to do. We want to be able to teach machines how to do that. We want to teach machines how to take digital pictures, start from pixels, and be able to do all the intelligent things that we do with our visual system. Answer all of those questions based on this type of input. Okay, so there have been many computational models of visual recognition. And I'm going to summarize them here in a somewhat succinct way by pointing out what I think are 6 of the fundamental properties of visual recognition models. Visual recognition models are hierarchical, that is deep in the current jargon. People refer to deep neural networks, networks that have multiple layers. Essentially, the visual system is using a divide and conquer strategy. So you're not trying to go from pixels all the way to a representation. You're trying to do that in multiple smaller and smaller steps. Okay, so this is, you divide a very hard problem that is going from pixels all the way to interpretation of the world, and you divide that into many smaller problems, each one of which can be done by very simple computations. There are convolutional layers, that is the same filtering operation is applied throughout the visual field. So you may have orientation tuning, and you may have neurons that are tuned to a particular orientation throughout the entire visual field. You may have neurons that are detecting a face, or a Jennifer Aniston, or a chair, and you may have those tiling the visual field so that the same computation, the same filtering mechanism happens in multiple different locations. There's a pooling or max-like operation to be able to achieve invariance to transformations. I mentioned at the physiological level this increase in receptive field sizes, and this is one way of achieving invariance to the exact position of a preferred feature in the visual field. So there's an increase in the receptive field sizes along the visual hierarchy. So as you go from one layer to the next, their set field size increase. There's also an increase in the degree of selectivity and the degree of tolerance transformations throughout these different layers of computational models. So people have been building these type of models for a long time. And about 6-7 years ago, there was a major transformation in the capacity of these models to do real world, to solve many real world object recognition challenges. So this came about in the form of deep convolutional networks. And there were three main principles, three main themes that the new era of computer vision capitalized on. One is these biologically inspired principles that I already alluded to, mapping the physiological responses onto computational architectures. Of course, these models benefited from major increase in computational power, our ability to access large computer clusters to do lots of computations, the advent of GPUs to do faster and faster computations. And then the other thing that played a transformative role is the enormous amount of access to digital images and to annotated digital images. So every time you go into your into Facebook or Instagram or whatever it is that you're doing, or you're tagging a picture, you're helping all the vision community by providing training data. So there has been an explosion in the availability of data sets that can be used to train computer vision algorithms. So one of the earliest one was MNIST. MNIST has become the equivalent of the hello world in the machine learning community. This is a database of handwritten digits that people use to start learning about deep convolutional networks and to start learning about machine learning. The idea is that you get images like these, and you need to train the algorithm. You need to train your vision algorithm to say this is a 0, this is a 2, etc. This is another very well-known database called ImageNet. There are 1,000 different categories. There are dogs and cats, actually many different types of dogs, many different types of cats. There are chairs, there are tables. There are 1,000 different categories and about 1,000 or more different exemplars for each one of those categories. So in 2012, this was considered to be a large number. So it was about 1 million, 10 million different pictures. The number of pictures being uploaded into the internet is growing exponentially. So essentially, we have a huge availability of training data. This is another one where not only do we have now pictures and labels, we also have tracings here indicating exactly the location of those objects and the shape of those objects. And these are very useful for many computer vision algorithms to train in terms of object detection. So before these deep convolutional network architectures, a lot of the approaches in computer vision were based on handcrafted features. So you started with pixels, and you spent a lot of time thinking about, and a lot of papers being published about, how can we extract good, useful features about those images. So you could think about, well, it would be useful to extract edges. Edges seem to make sense. So how about if we extract edges? How about if we extract colors? How about if we extract different varieties of shape properties? And then all of that is conveyed to a system that learns a readout mechanism and learns to map those features into labels. So they say these features are present in chairs but not in tables, etc. But then what people started doing, and that's what most computer vision algorithms do nowadays, is what's called essentially end-to-end training. Basically, you start with pixels. At the end of the day, you have labels, and you want to train the whole system. That is, you want to learn all those features. Instead of handcrafting those features, you want to learn all those features from the data. And in order to learn that, most of those, at the heart of many of those algorithms, there is a procedure called backpropagation, which essentially entails taking the output of the network, comparing that with the desired label, and if there is a discrepancy between the two, if there is an error between the two, that error is propagated backward through the entire cascade of operations in the network. And by calculating the gradient, how much the loss function, that is the error, the difference between the target and the actual label, depends on the actual activations and therefore on the actual weights, which are the knobs that you are manipulating in the network. So by doing that, people started building deep convolutional networks like this one to a very coarse first approximation. We can map these type of structures onto this hierarchy of the visual system. So essentially, what you're seeing here is pixels. At the end of the day, you have this vector of 1,000 numbers, which tells you this picture contains a chair, this picture contains a table, et cetera, et cetera. And this goes through a cascade of computations that are based on those essential ingredients that I mentioned before: filtering, pooling, normalization, and in particular, these filtering operations are done in this convolutional manner throughout the entire image. So roughly speaking, we can think of these the pixels as the retina, and each one of those stages, people imagine that they map somehow onto these boxes here. Of course, there is as you can see already from this diagram here, there is much more complexity here than here. And actually, if you peek inside of these boxes, there is a world of exciting biology that's happening here that's not incorporated into these computational models, and that I suspect will lead to even better algorithms and the next generation of computational algorithms once we begin to dissect in higher resolution what's happening at the biological level. One of the races in the computer vision community was to build deeper and deeper networks, so adding more and more and more layers to get better and better performance. This was 2012. These are the error rates that they were getting in 1,000 way classification. So because there are 1,000 categories, chance would be 1 in 1,000, so 0.1% is what you should get as chance. So your error rate, if you're randomly picking labels, your error rate should be 99.9%. If you're doing top 1 classification, that is only one label. Okay? If you're doing top 5 classification, that is sometimes in the computer vision community, people refer to a particular label as being correct if any of the top 5 labels are correct. Then if you're doing top 5 classification, then your error should be 99.5. But these are the errors that they were getting in 2012. So it's quite impressive. And this has steadily improved over the years by building deeper and deeper network architectures. In addition to getting better and better performance, it turns out that these algorithms also can capture essential aspects of the variance in the responses of neurons in the brain. So what I'm showing you here are 3 example neurons. These are recordings from Jim DiCarlo at MIT. These are recordings from this area, inferior temporal cortex. This is the same area that I mentioned earlier that has neurons that are selective for complex shapes. And what this diagram shows here is a large number of pictures. So each one of these numbers corresponds to one picture. So this is this picture here. This one is this picture here. This is this picture here, and so on. And then on the y-axis, you have the firing rate. That is how much activation how many spikes were triggered by this picture for this particular neuron. So there are 2 traces here, the black and the red one here. So one of them corresponds to the activity of the neuron. The other one corresponds to the prediction of the firing rate based on these computational models. So you can take one of these deep convolutional network architectures. You can pass this picture onto a network, and based on the activation pattern of the computational model, you can predict reasonably well how the neurons are going to respond to all of these different images. Furthermore, there's a correlation between how well you can explain biology, how well you can explain neurons, and how well these algorithms perform in real-world object recognition tasks of the flavor of image classification in ImageNet. So what you're seeing here now is categorization performance. So one would be if you can classify every image on the web, on ImageNet correctly. And then on the y-axis, what you have here is the fraction of the variance in the responses of neurons in inferior temporal cortex. So here, 100% would be if you can explain the responses to every single picture. Each dot here, each point here now corresponds to a different model. So these are different deep convolutional architectures or different models trying to either do object classification or trying to explain biology. And the point that I'm trying to make here is that there is an approximate correlation between these two worlds. That is, models that are better at explaining biology are also better at object classification on average. Some models are very bad. For example, pixels are very bad. So if you're just trying to do one-step recognition purely based on pixels, it's a very, very hard problem. Okay, so this is like going back to those numbers I show you there and try to figure out that that's a flower purely from one single layer or one single readout purely based on pixels. It's very hard to do that. Models do pretty poorly at doing that. That's not a very good model for biology either. Okay, and then at the top here, we have better and better models that can succeed in this world and in this world. As you may see from the numbers, we're still explaining only a fraction of the variance in the responses in IT. So there's still a lot about biology that we don't understand, and we need to understand better. The point I wanted to make here is that there is an approximate correlation between these two worlds. The better we understand biology, the better models that we can build for computer vision. I want to spend the next yes. So this is a model that basically it's doing just categorization based on category labels without necessarily using the pixel observation. So it's a kind of a psychological model. It's different from this should not this model knows exactly what category it is. But if you just use categories, of course, by definition, you can solve this categorization of ImageNet, but that doesn't explain biology at all. So neurons don't seem to reflect the semantic categories such as dogs and chairs and tables. It's not that neurons that you can use that as a way of explaining the responses of biological neurons.
How do they map the model responses to firing rate? Because normally the models just give labels.
Right. So what they're doing here is let's take these models. So there are multiple models here, but the basic idea is you start with a picture. Okay. So this is the picture. The picture goes through this cascade of operations, and then you stop somewhere at one of these layers. Okay. So let's stop here, for example. So at this level, I have a bunch of numbers that are representing that image. Okay. So I have depending on which layer we are in. We have many features, many shape features by which this model represents that particular picture. So now once we have that, I can ask, how well can I predict the firing rate of the neuron based on these numbers? I can correlate these numbers here. These are the features that the model uses to represent that picture with the firing rates. Okay. Of course, you need to do cross-validation to ensure that the training set and the test set is separated. So you take a lot of pictures. You have a lot of these vectors. You have a lot of firing rates. You build a map between those two. In this particular case, they're using a pretty simple linear mapping between that and the other, between these features and the firing rates. And that's how you try to explain firing rates. And then I didn't get into the details here, but you can also ask questions about which of these levels better explains the responses in a particular brain area. So I sort of here. I sort of alluded to the idea that these approximately maps to these approximately maps to these, and you were all very generous and you didn't question that, and why do I think that this box here maps onto that one? So one of the reasons why we think that this box here maps onto here is that when you do these exercises, it turns out that these units here are a little bit better at predicting responses here than there. And these boxes here are a little bit better at predicting these responses than those responses. Okay, and that's part of the motivation for these arrows here.
So do the algorithms actually have the same hierarchy compared to the brain? So is the mapping, like, do the mapping not overlap at any point? Or are they like actually just like that?
These arrows are only an approximation. So I use PowerPoint to draw these arrows. There's nothing more sophisticated than that. Now, the data behind this is this type of correlations that I was just mentioning. So you can ask, how well can I explain responses here based on this activity? How well can I? So if I take the activity of the units in this particular model at this layer, it turns out that we can explain this better than that, and so on. Okay, so there's an approximate mapping, but it's not perfect. And so to answer your question rigorously, I would have to go into each model. And it's conceivable, and in fact it happens, that I think what you're saying is that in some cases this might explain this one better, and this one might explain that one better. And that might happen. In general, there's an approximate monotonic mapping between the two. But again, this is only very coarse. There's a lot more than we need to do to map these two worlds. And in fact, on purpose, I used this model from 2012 because the number of layers here is approximately similar to the number of boxes here. But if you look at more recent instantiations, you quickly run into trouble because here there may be 100 different layers. And then we have to say, well, how do those 100 layers map onto these? So then we need to start talking about the fact that each of these boxes itself have multiple layers inside it, and so on. So this should be taken with a grain of salt. There's an approximate correspondence between biology and deep convolutional network structures. And there are reasons to believe that we can push both fields simultaneously. And that we can use biology as an inspiration for these models. But I don't really mean to say by any means that this is a one-to-one and only mapping. Definitely not. Okay. Yes?
Are there any models today that go beyond just labeling objects and perhaps saying what's going on in the image or
That's a very good question. I want to spend the last couple of minutes flashing very quickly a couple of other things that people do with these models. I think it will partly answer your question, but if I didn't, ask me again. Okay? So labeling objects has been one of the main things that these models have done. There are a couple of other things that people have tried to do with this type of architectures, and as I alluded to at the beginning, there's much more to vision than just saying, is there a chair, yes or no? So this is one of those things. This is called deep dreaming. The idea is that now, if I have one of these models, I can look at the activity of one of these units in the model. Essentially, I can stick an electrode in the model. I can record the activity of a unit in the model. And I can ask, what kind of pictures will elicit high activation in this unit? So this is one of the units. Again, I'm working here with the AlexNet network. This is the network I just showed you. And we're looking at one particular unit at the very top. This is unit number 209. This is a unit that's involved in saying, is there a Labrador picture, yes or no? Okay? So when we present pictures of different Labradors, you get higher. This is the activation of this unit. You get higher activation. When you present other pictures, you get lower activation. And now we can ask, well, that's fine. So all of these pictures from the internet, from Labradors, elicit high activation. The network was designed to do that. Now, can I create new images? Can I create images that will be exciting for the neuron? So this is the deep dreaming algorithm. The deep dreaming algorithm is trying to generate images that will lead to high activation. For this particular image here, the activation is here. So this image here, this image here elicits much higher activation than any picture of a Labrador in the web, even though this is a Labrador unit in the sense of discriminating whether the Labrador is present. In other words, the way the people who made this model try to sell it is, this is what the neuron dreams about. This is what really excites this particular unit in the model. If you squint, you can sort of see multiple Labrador dogs kind of in there. Here's another example. This is a unit that's involved in labeling objects as whether there's a desk or not. It's more strongly activated for desks. And when you do the deep dreaming, you generate images like this one, which trigger higher activation than any image available on the web to date. So another thing that people have done, and that's become quite fashionable in the last couple of years, is to use what are called generative adversarial networks. And those are networks that are trying to create images, essentially to deceive some of these networks. So you have real images, you have another system that's trying to generate pictures, and you're trying to essentially you have a discriminator network, and you're trying to generate images that this network will not be able to distinguish whether they are real or not. And this has been used in a large number of applications. Some of you may be familiar with some of these. These are, for example, trying to generate images that look like or follow a particular model. So, for example, you can take this picture. Many of you may recognize it. I think it's from the Avatars. What's the movie? Anyway, so this is from a famous movie. You can take the Gioconda here, and then you can render the Gioconda based on these features. Or you can render this particular avatar, I forgot her name, based on the features of the Gioconda. So basically here you're generating images in a particular style based on this idea of trying to generate images that will deceive these networks. Another thing that you can do, this is the work of a grad student, Bill Lotter, is trying to build networks that will predict the next frame in video sequences. So you're not just doing object classification, you have a video, and you're trying to predict the next frame. So essentially you're trying to ask what's going to happen next. So these are actual videos, and these are actually predictions. So the network is generating images, it's predicting what's going to be the next frame. If you pay close attention, you'll see that there's some difference between the two, but it's pretty good. So you can predict what the next frame will look like based on these deep generative networks. And then this is another example. We can use also these networks to predict the patterns of eye movements, where people are going to be moving their eyes and how people are going to explore the different images. If anybody's interested, I'll put all the references. You can go in depth in more detail. I want to come back quickly to this very good question that was asked, and very quickly mention that we're still very, very far from extracting meaning and being able to answer an arbitrary number of questions. So there are systems out there that try to do image captioning. This is one of them. It's called CaptionBot. You can go online and play with it. You can upload your favorite pictures and see what the system does. And very quickly, you can make the system fail and laugh at how ridiculous the statements are by the system. So if you put this picture here, the system says, I think it's a group of people standing next to a man in a suit and tie. So it's actually pretty good. I was actually quite surprised. So it's detecting that there are people here. It's detecting that they're standing. It's detecting suits and ties and so on. Of course, I would argue that the system is still very far from understanding everything that you can see about this image. This image is meant to be somewhat humorous. Obama is being playful. It's stepping on these things. Some of you may be too young to know, but this is a scale. So people are self-conscious about their weight. So Obama is playing with, is exerting a force, and therefore making him think that he's fatter than he actually is, and so on. So there's a lot more to this picture than this. And yet, so this gives you an idea of the state of the art in sort of what you refer to as understanding. So I think we're still very, very far from understanding images. But if you play with this will give you a sense for state of the art in image captioning. If you play a game like, for example, decomposing the image and doing this, so all the fun of the image goes away, a lot of the meaning of the image goes away, and yet the answer from this system is exactly the same. Again, suggesting that we still have a long way to go to really understand vision and build intelligent systems. So I'm going to stop here. I'm happy to take more questions. If people need to go, they can approach, and I'm happy to continue conversation. Next Tuesday, we'll have our first invited speaker, which is going to be Chaim Sompolinsky, one of the world leaders in computational neuroscience. So thank you.